move on to the next talk, which is from Jeremy Mirza. Jeremy is a postdoc working on the CADE project at the University of Birmingham. And he is going to give us an overview of a recently released protocol for automating SARS-CoV-2 sequencing on the popular and inexpensive OpenTrons platform. So over to you, Jeremy.
Hi, Nick. Hi, everyone. Can you hear me properly?
We can hear you. Just need you to share your screen now.
Perfect. Hi. So, as Nick said, my name is Jeremy Mirza. I am a postdoc in Professor Lohman's lab, and my project has admittedly been primarily focused on working on studying arboviruses within the CADE project, which I think some of our collaborators from Brazil are on this call on. And obviously with the current pandemic and everything that's been going on, we have moved and sort of adapted a lot of this for a number of other protocols related to COVID-19, such as the one that Josh has been discussing previously. And the main aim of my research one of the primary aims has been to sort of automate this process in a way that can be slightly more affordable or attainable, and hopefully is attainable for some of the people on this call. And it's quite topical in a way that it's incredibly important to have some form of improving or making the protocols themselves a lot easier. Josh already mentioned how it would be nice to make this protocol, especially for Oxford Nanopore, to have a reduced number of steps, or to make it more accessible and more user-friendly. One of the main reasons for that is obviously, as this is a protocol involving amplicon sequencing, there is a high chance of contamination. In fact, one of the recent bits of news that I'm sure a lot of you have already heard about is this new sort of scary variant allegedly discovered called Deltacron. However, when we look at the actual tree formed from the GISAID repositories, these are all highlighted in red. This was sent to me by Tom Peacock. You can see there is no sort of direct link. There's no sort of actual clustering, which is very emblematic of what you get from contamination. To give you an idea for those interested, this is what an actual cluster would look like. This is a recombinant form of alpha and delta, which was discovered by a group in Japan last year. So there are several ways to mitigate for this. One of the most notable being, as Josh mentioned, sort of very good lab discipline, making sure you separate your sort of clean environment away from your amplicon containing environment. And another thing to help with this, especially as you're upscaling and going through large numbers of genomes, as Nick mentioned, there's over 7 million now that have been published on GISAID, that are now available. The important thing is to sort of how to maintain a consistency as you are upscaling. And the easiest way is to use some form of liquid handling robot. Now, admittedly, certain robots, such as those made by Tecan and Hamilton, can admittedly have quite a high degree of difficulty for the user in terms of just getting accustomed to it. The UI can be a little bit alien when you first start using them. And also they can be a bit prohibitively expensive for a number of labs. One of the other issues as well is that you can have a certain amount of labware that is incompatible, and you will need to buy it from certain suppliers, and this may not be available if everyone else is trying to acquire the same labware. So as if one of the issues in itself, so the advantage with OpenTrons is that a lot of these issues are completely mitigated for. It gets around it is a lot cheaper as a robot. This is primarily because of the way it utilizes liquid level detection, in that it basically relies on the user to manually calibrate the robot rather than using conductive tips, as you would see for Tekken and Hamilton. The other thing that is a lot easier with this robot is the way it is set up for how you design and write protocols. So they have two options. One of them is this thing called a protocol designer, which I can post a link to in the chat if anyone is interested in just playing around with it. And essentially it will show the layout of the robot, which will have these 11 positions. And on each position, you can put a particular type of labware, be it a particular type of plate, a reagent reservoir, or a set of tips. And from that, you can then input and enter the different instructions for the protocol, as well as entering liquid classes. For those who are more comfortable with liquid handling robots, you can use something called the Python API. So what that allows for is essentially a similar function to the protocol designer. However, it has a lot more complexity and there are a lot more complex commands that can be done. Such an example includes, so the way this would work, for example, is that you have 5 sections. One is the one part is the metadata, where you define the sort of general parameters for the run. You just give a basic description, who the author is, and the API level or the version of the protocol you're using. The entire run itself and all of the commands used are defined within a function. And then within that function, you will set what the labware is. So at each position, you will basically designate a type of labware. And the OpenTrons API has a number of types of labware that are predetermined. So it already knows the parameters for, it already has a base calibration settings for, so it can recognize these. I'll show you something a bit later, how we're able to use this and manipulate this when using the robot. And then after that, you will define what pipettes you're using and then the actual run itself. So within this course and within the sort of CADE project, I've designed several protocols, primarily for COVID-19 sequencing, both for nanopore and Illumina chemistry. As these are the sort of most standardized protocols utilized for this. But within this as well, we've also designed some very generic DNA bead cleanups, as you would use for the Illumina sequencing protocol, as well as very standard sort of rapid and native barcoding for nanopore technologies. I'm also working on a couple of other protocols that we still need to validate, admittedly, given the number of tips that you can go through during some of these protocols. One of the nice things about the tip boxes for OpenTrons is that each position is its own sort of compartment. So in theory, you can actually use the same tip between multiple steps without the risk of sort of cross-contamination of other parts of the process. And the other thing you can use is, if you're working on a primer scheme, such as the ARTIC one, as Josh mentioned, you can be working with 100 or up to 200 oligos if you count the forward and reverse primers. And when you are making this up, it can be quite a laborious task for one person to do. So having, if you use the robot and import an Excel sheet with all of the variables needed for all of the dilution factors, this is an option as well. But the main protocol I've been working on is for the low-cost assay for Oxford Nanopore Technologies, which is a process that involves sort of 2 general sections: a pre-PCR stage, where you will extract the RNA from the primary samples, and then prepare the cDNA, and then amplify this with a PCR reaction. For this, at the moment, I still maintain this as a manual processing step, predominantly because in a lot of cases, we will have RNA samples that are stored sort of separately or require sort of manual manipulation. And admittedly, it is possible to run this stage or an automated process as well, but the main focus has really been on the post-amplification steps, which are done in a separate lab to avoid amplicon contamination. So for this, it can be roughly broken down into about 8 steps. And then for this procedure, I tend to automate the first 4 stages. And then once you get to the PCR or the pooling of the libraries, you end up, it's actually becomes easier to finalize the process manually. So what I'll do now is I'll just go through some of the protocol, and I can just show you sort of some of the features of the robot and how you can trick the robot essentially into working for you. So one of the nice things is that the way we can use this robot is we can calibrate it from the base of the labware. So what that means is the Z position or the Z max position is always going to be the lowest position that you will use. So from that means you can get very accurate pipetting even when you're using custom labware, because according to this robot, it actually thinks that both of these are deep well plates, but because I've calibrated them separately, it will be able to utilize the same movements without having any risk of collision. One of the other nice things as well is if you, because of the open source nature of OpenTrons, we're able to use a lot of custom labware designs, including this glorious plastic tat that I have here. And with that, we're able to basically customize and modify the robot to fit each lab that we're hoping to work with. We've done this a lot with our Brazilian collaborators as well. One of the other nice things with this robot is while I have a multi-channel head attached on both of the positions here and here, you can actually use these as a single channel as well. So in the API, what I've written here for this function is that you move from position A1. So you always use row A for a lot of the transfers. However, if you put, for example, position H, you will only pick up 1 tip. And if you put position G, for example, you will pick up 2 tips. Using this, we can basically manipulate the robot to function in a way where it thinks that it will act as a single channel pipette. So if you are transferring a large volume of samples to a single pipette or vice versa. So for example, in this case, when you have a much more valuable master mix, such as the ligase enzymes or the end repair enzymes that we use, it means that your sort of expenditure compared to if you would use a reagent trough is a lot more reduced. Another thing we have for the next stage of the process, which is the barcoding of the plate following on from when you perform the end preparation, is that you can set it so you can have both of these plates be the exact same thing and be the same dimensions. However, if you want, for example, a lot more precise and accurate pipetting, which especially may be the case if you've got a requirement of about only transferring, say, 1 microliter or even less than that, then by calibrating these individually, you can essentially, sorry, excuse me, you can maintain accurate pipetting. The final thing as well, to go through the final stage of the process, is when you're actually pooling your libraries together. For different people, they may use 96 samples per a single flow cell. For others, they might use 48. So for this, we can do something very simple. So we can maintain what is essentially the exact same protocol and the same script, but we can condense a large part of the script here into 5 positions rather than 11 in this case. So I have admittedly gone through a lot of that very quickly, but it was just to sort of give you a general idea of how this protocol works. For those of you who are interested in potentially trying this protocol, if you already have this robot, if you are interested in buying this robot, for example, I tried as best as I could to make sure this didn't sound like an infomercial for OpenTrons, and probably failed. But I have a very simple GitHub repository, which will have a version of the protocol up online. And this is forked directly from the previous protocol designed by Josh. And so I will post that link in the comments as well, if you're interested. Just as a final thing, I'd like to thank CADE for funding this project, and especially Professor Esther Sabino for inviting me to her lab, and everyone in her lab. And hopefully some of the Brazilians are already awake and on this call, just to say hi, but for all of their help in testing and troubleshooting this protocol. Thank you very much.
Brilliant, thank you, Jeremy. That was a really nice introduction to OpenTrons. And as you say, they haven't paid us for that presentation. We were just enthusiastic about the idea of being able to purchase a robot relatively inexpensively. Can probably tell us how much it costs, and also something that you can get up and running without too many visits from the engineers and without having to take out expensive support contracts. They're very attractive to us. So we have one of those in Birmingham, and as you said, you set one up in Sao Paulo as well. Now, how much approximately do they cost?
So the base model for the actual robot itself, plus the 2 pipettes, will cost around £8,000. I'm not quite sure of the conversion, and obviously things like VAT or the equivalent in each country will be different. But an equivalent robot, from example from Tecan, will cost upwards of, say, £80,000 to £90,000 at the moment. So relatively, for most users, you can gain the same functionality out of something, which is thankfully a lot cheaper. And as you also said, doesn't require a very, very expensive engineering contract as our Hamilton does.
Yeah.
So,
yeah, and that's about $11,000 at the moment. And although that's not cheap by any means, actually, if you're going to do sequencing at any scale, you will spend a lot more money than that on sequencing reagents and consumables. And therefore, anything that increases the reliability and the quality of the data is probably a worthwhile investment if there's going to be sequencing at scale being done. So we have some questions in the chat, which is great. Artem has 2 questions. Does OpenTrons use some kind of special pipette tip? Or are they similar to those used for manual pipettes? And also, can OpenTrons be adopted for working with 384 well plates?
So OpenTrons provides their own sort of form of pipettes, which are already recognized by their API, but functionally, these are the same as your standard tips made by Biorad, by Biofill, by any of these companies. So there's nothing special about the tips themselves. If you want to use your own type of tips, it is entirely possible. We've set up the machine in Brazil, in Sao Paulo, to use a variety of different tips from different companies. The one important thing is to make sure the box itself will actually fit into each position. So for that, you can, we've designed a couple of simple 3D printed adapters that will allow the box to fit in consistently, so that there's lower risk of miscalibration or not picking up the tip successfully. And then for the second question, yes, I've designed the COVID-seq protocol to work within 384 plates as well. But the manner in which I set it up is that you work with, so generally with those, if you're working with a 384 thermocycler, I try and keep the majority of processes in 4 separate 96 well plates during the procedure, just to minimize the risk of collisions. However, it does have the accuracy, and it does have several 384 plates already recognized by the API. So yes, it is possible. Okay, excellent. There are 2 questions. One is from Li Jing about saying, based on the fact that deltacron was likely generated from contamination, do you have any idea what proportion of sequences submitted to GISAID are contaminated? That seems like a difficult question, but do we have any sense of that? Off the top of my head, honestly, I wouldn't be sure. Again, is a lot of the ways I would pick up contamination, and a lot of the ways I would test for contamination when I was troubleshooting this robot was actually to go into the BAM files themselves and look to see if I'm getting sort of cross-contamination, or if I'm seeing sort of these kind of a multiplicity of reads where I'm expecting where a particular part of the sequence has always been consistently something for one variant or another. But ultimately, it's from my perspective, it's difficult to say. However, one way you could potentially analyze this is in the same way that Tom Peacock and I think a couple of others did on Twitter with this recent variant from Cyprus, which would be to take supposedly very similar sequences that are purportedly from a a single cluster and then run them through this and see how they compare phylogenetically to the majority of COVID-19 sequences.
Yeah. I mean, in the next session, we're going to cover QC in some detail, and there will be some examples of how you can detect these kind of problems from Jared Simpson, and also some other ways of mitigating these problems from Catherine Siddle. So we'll keep picking up on this. So there are actually a few more questions that are quite technical in the chat. In the interest of time, I'm going to ask Jeremy if you can answer those in the chat so that we can keep this session to time, which is due to finish at midday in about half an hour.